In what seems like the plot from a science fiction novel, researchers built a new windpipe for an Eritrean man who had a rare but deadly form of tracheal cancer. Professor Paolo Macchiarini of Karolinska University Hospital in Stockholm performed the surgery with the help of an international team, including Boston-based Harvard Bioscience, which designed and built the bioreactor used to seed the patient's stem cells onto a scaffold.
The synthetic scaffold was made in about 2 or 3 days in the laboratory of Professor Alex Safalian at University College in London. And it was mated to the bioreactor, which we built. The bioreactor is really the box, the chamber in which the cells are slowly deposited onto the surface of the scaffold, which was made by Professor Safalian. That process also took about 2 days of growing time. It took us a similar period of time to Professor Safalian to actually make the bioreactor. All of this was done under extreme time pressure, I might add. This is not normal for anyone to make these kind of things in this short a period of time, but in this case, there was no choice. The man had a very short period of time in which the operation might be successful.
Although human tissue has been successfully generated outside the body, Harvard Bioscience's David Green says the trachea transplant is a first.
Materials, other research groups, other clinical groups have produced expansions for bladders, for patients who are born congenitally with very small bladders. That's not really a transplant so much as it is more of an augment or an expansion of the existing bladder. And it's also been done for ureters, which is the tube that connects the kidneys to the bladder. So there are other examples of organs being generated, or at least tissue being generated outside the body. I think this one, the trachea, really was the first full organ that was generated outside the body.
Carl Simon of the National Institute of Standards and Technology points out another unique aspect of the landmark surgery.
Previously, people would take a cadaver and take a body part out of that and then remove all the immunogenic material from it and then seed that with cells and grow a new tissue. Macchiarini's group has used fully synthetic material, so it doesn't require a cadaver or a donor. Of course, there's always a shortage of donors. You have problems with disease transmission. The cost, if you take a donor material, you have to do a lot of testing to make sure it's safe to an implant, and there's a lot of variability person to person, donor to donor.
Simon is with the Biomaterials Group at NIST in Washington. He says the shape of the trachea was created with a flexible man-made scaffold.
Macchiarini's group used this type of scaffold, which is a nanofiber scaffold. These ones have become very popular recently, and a lot of advances have been made with these. The reason is because they mimic the structure of your natural tissues.
The role of NIST in tissue engineering is to provide a platform of reference materials to help the industry thrive and grow. So the NIST laboratory is equipped with its own nanofiber making machine and racks of stem cells that are kept frozen in liquid nitrogen, making it able to replicate some of the research into tissue generation.
So these are some images of human bone marrow stromal cells cultured on nanofibers that we took here at NIST. The red staining is a protein in the cell body called actin. The nucleus are those yellow dots, and these ones are being cultured on a nanofiber scaffold. So you can see what these cells look like, and these are the same cells that Macchiarini's group used, but Macchiarini's group took these cells from the patient's bone marrow, and then they differentiated them into chondrocytes, cartilage cells.
The process, as described by Green, seems nothing short of miraculous. Just a few days before the surgery, bone marrow was taken from the patient's hip bone. From that, about 40 milliliters of stem cell material were bathed over the surface of the very porous scaffold.
So as the stem cells are poured over the surface of the scaffold, the individual cells settle into those porous holes in the scaffold material, and it really feels like home to them. And then they start multiplying and dividing and start joining up with each other. And after about 2 days, they've joined into each other, and they've become more like tissue than like just cells.
Both Green and Simon agree the possibilities for this technology are endless. Research is underway already for generating more complex organs like hearts, kidneys, and lungs. While such solid organ transplants may be years away, Green says the next stage is to expand the technology to other hollow organs like ureters, blood vessels, and coronary arteries for bypass surgery. Rebecca Ward, VOA News.